Hello everyone. Today I'm going to talk about the 2025 Nobel Prize in Physiology or Medicine. On October 6, just recently, the Nobel Assembly awarded the Nobel Prize to three distinguished scientists: Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi, for their discoveries concerning peripheral immune tolerance. But before moving straight into it, let's talk about the immune system first. It is a complex network of cells, tissues, organs, and proteins that defend the body against infection and disease by identifying and destroying foreign invaders like bacteria, viruses, and parasites, as well as abnormal cells such as cancer cells. To simplify, if each human body is a country, there are more than 37 trillion human cells living in this entire country. Within it, there is an army consisting of 1.8 trillion immune cells, protecting against foreign invaders and cells living inside which lacks discipline. It is a substantial job as they work tirelessly trying to identify and neutralize harmful pathogens within the 60,000 germ types a person encounters every day. On the other hand, they also have to sort through billions of cells experiencing mutations, of which a very small percentage become cancer cells. These mutated cells can imitate normal cells to evade the immune system, such as changing surface protein on cancer cells so that it can hide. Pathogens can also imitate through molecular mimicry, producing proteins that are structurally similar to host protein, basically camouflaging themselves. Then, how complex and accurate should our immune system be so that it can neither allow any foreign enemies to invade nor internal chaos to occur, especially not mistakenly killing innocent cells? That is what the three Nobel Prize laureates discovered. The laureates identified the immune system's security guards, regulatory T cells, which prevent immune cells from attacking our own body. Shimon Sakaguchi was swimming against the tide in 1995 when he made the first key discovery. At the time, many researchers were convinced that immune tolerance not only developed due to potentially harmful immune cells being eliminated in the thymus through a process called central tolerance. The soldiers of immune cells must be trained in a special boot camp in the thymus for the T cells and bone marrow for B cells. During this training, they learn to tell the difference between their own body cells and invaders. If a cell reacts strongly to your own body proteins, it is very dangerous, so the body reprograms or gets rid of it. This process where the immune system learns not to attack itself is central tolerance. Yet this is a misconception because it only removes most but not all self-reactive cells. This is why the body has peripheral tolerance also, a backup system which Sakaguchi discovered in 1995. He identified a previously unknown subset of T cells, known as regulatory T cells, or Tregs. Among the graduates of the boot camp, there are the special police officers. Instead of fighting criminals or pathogens, they suppress self-reactive immune cells that aren't identified in cell tolerance. So peripheral tolerance happens in the real world after training. Tregs control the immune response to self and antigens to prevent autoimmune disease. There are diverse methods to restrict these malfunctioning T cells, such as secreting inhibitory cytokines, direct cell-to-cell contact, and metabolic disruption. Sakaguchi identified the protein CD25 in 1995, helping to distinguish and isolate this class of T cells. To clarify, CD25 isn't the only key marker for Tregs, as there are other biomarkers such as CD4 and FOXP3 used more precisely to identify them. So this is his experiment. Firstly, Sakaguchi and his colleagues observed that mice that had their thymus removed at birth developed various autoimmune diseases. They then demonstrated that they could prevent those autoimmune diseases by injecting a specific fraction of immune cells from the normal healthy mice into the mice. This suggested the existence of a T cell population that could suppress the immune system. To identify this population, Sakaguchi spent over a decade trying to differentiate this new class of cells from other cells. Sakaguchi demonstrated that these suppressive T cells could be identified by the presence of both CD4 and a specific cell surface protein called CD25. This new class of cells was named regulatory T cells, or Tregs. Subsequent research validated this discovery by showing that mice lacking the FOXP3 gene spontaneously developed fatal autoimmune conditions as their Tregs were not functional. This is the first half of the whole story, where he discovered the cell type of a specific group of T cells that help in peripheral tolerance. On the other hand, Brunkow and Ramsdell were working at a biotech company called Celltex Cryoscience in the 1990s and had a particular interest in a mouse strain called scurfy. In these mice, the males were born with a scaly, flaky skin, enlarged spleen and lymph glands, and a lifespan of only a few weeks. It was known that scurfy was linked to the X chromosome and was caused by the T cells destroying the mice tissues. But Brunkow and Ramsdell want to understand the molecular mechanism underlying the disease. This required mapping the entire X chromosome in detail, narrowing it down to 20 potential genes. Eventually, they discovered the faulty gene responsible for scurfy, and due to its similarities with FOX genes, called it FOXP3, which destroyed immune balance, as the mice had no functional Tregs. They went on to discover a human version of the FOXP3 gene, which was mutated in individuals with the X chromosome-linked autoimmune condition IPEX, and in 2001, they published a paper in Natural Genetics revealing that mutations in FOXP3 gene cause both IPEX in human and scurfy mice. Researchers went on to make the link between this discovery and Sakaguchi's work on regulatory T cells, and 2 years later, Sakaguchi, followed by others, proved that the FOXP3 gene controls the development of regulatory T cells. Well, in the end, why do they deserve the Nobel Prize? They had led to the development of many new treatments, and there are now over 200 clinical trials involving regulatory T cells. Their discoveries are important as they explain human diseases. They unified central and peripheral tolerance and completed the puzzle of immune self-recognition. In summary, they revealed the cellular and genetic basics of immune tolerance, discovering the peacekeeping T cells, Tregs, and their master gene FOXP3. These great scientists totally transformed immunology, medicine, and our understanding of how the body avoids attacking itself. A discovery fully worthy of a Nobel Prize. Well, that's all for today. Hope you enjoyed this dive into biology. Don't stop exploring the world around you, and I'll see you next time.